So good evening, everyone. Hello, James. Hello, Saul. It's a pleasure, James, to have you at my channel tonight. So I decided to talk a little bit about impurity of molecules, and I think that this is a problem for prebiotic synthesis of the first life forms, and I brought it up in one of the streams in a few days, and then Leophilios said, oh no, that's not the problem. So I actually made a little bit of investigation today about this, but I didn't actually find so much literature I didn't have much time either to figure out what science papers say about this problem. So James, maybe you could give a word in regards of that. What do you think? Is that the problem or not?
Absolutely, it's a problem because it leads to messy chemistry, and you have to remember we do have very strong evidence that in these primordial types of conditions, the number of molecules that have nothing to do with life, that are useless to life, or very often detrimental to life because they will compete for reactions. They're sticky molecules that will attach themselves to those crucial molecules needed for life. It'll disrupt membranes, it'll disrupt polymerization. They will attach themselves to DNA, to RNA, to protein, and they outnumber the good molecules, which we call the biotic molecules, the molecules that we find in living organisms. These abiotic molecules, these ones that are either not beneficial and most likely detrimental, by far outnumber the biotic molecules. When we look at the Miller-Urey type experiments where they just start with some seed chemicals, you know, like ammonia, methane, hydrogen cyanide, hydrogen sulfide, water, things like that, and let them just react with some, you know, electrical zaps as a source of energy. The majority of what is made, you can go back and look at that Miller-Urey experiment, and they said that it was mostly, you know, tar, asphalt, you know, these complexes, these aggregates of molecules that all just kind of stick together into a bunch. And the other molecules they made were, well, you know, you have formaldehyde and hydrogen cyanide, which are two known poisons. You know, if you're generating cyanide, you know, that's a chemical weapon. Now it doesn't affect, it only affects cells with mitochondria. Or that have the electron transport chain, so bacteria as well. It disrupts the complex 4 in that chain. And formaldehyde we use in embalming fluid. It's a cross-linker. It makes the proteins cross-link, and nothing grows in it. That's why we use it to preserve specimens in it. But it is a seed material to make sugars abiotically, so it's necessarily there. But the glassware from those experiments were reanalyzed years later once we had better technology like mass spectrometry. And when they were run through these machines, they found that there was, you know, over 10,000 abiotic molecules and mostly aggregates for just a handful of crucial molecules, like a couple of amino acids and a few sugars, you know, the things that life needs to make DNA and proteins. And then we look at the meteorites, the carbonaceous meteorites, like the famous Murchison meteorite. I think it was found in 1969 in Australia. And about 1% of that meteorite was carbon material, organic material. They found dozens of different amino acids, you know, most of them not the kinds of amino acids that we use in life today to make protein. So they're non-proteinaceous amino acids. It's just a molecule that has an amino group containing a nitrogen and a carboxyl or carboxylic acid group on it as well. So any molecule that has both of those is an amino acid, but most of them that are produced abiotically are not used to make proteins. So you've got a problem there, because when you're trying to assemble a protein without ribosomes, you know, in this primordial condition, or in a protocell or something, it's just going to be a random, not even random arrangement. It's going to be selected by physical and chemical selectivity. So the amino acids that are nonpolar will tend to be linked up to each other, including the ones that we don't find in proteins today. And you also find them linked up based on how much the functional groups will crowd each other. So if there's a big bulky chemical group right here, and it's going to interfere with another bulky chemical group sticking off that amino acid, they're going to repel each other, and they won't attach to each other as much as, you know, a very simple amino acid like glycine, which just has a hydrogen as its functional group, or alanine, which just has a methyl group, a carbon and 3 hydrogens. So chemical selection is going to favor, you know, the more stable molecules, and the order of the amino acids is going to be based more on their relative abundance. You know, the higher, if some amino acids are made at higher concentrations than others, those are going to be the ones next in line to form these chains. And there's no biological or functional selection at that point. It's just, you might make a random protein that folds into garbage the vast majority of the time. Even when you take one of our proteins that we know works and you heat it up, you denature it, and you let it cool down again, more often than not, it won't fold back in the same exact 3-dimensional shape. Now take in the fact that you've got thousands and thousands of other molecules besides amino acids that are going to bind to this growing chain, whether it's a chain of nucleotides that are going to form RNA or DNA, or a chain of amino acids to form a protein, polypeptide. These polymers, as they're being assembled, are going to have their reactions stopped all the time. Let's say you have, you know, these amino acids have sort of a left side of them and a right side. So the amino group and the carboxyl group. Well, what if the carboxyl group came off? What if it got oxidized to CO2 and came off of there? So now it's like trying to form a chain of people, but someone came in who's an amputee and is missing one of their arms. There goes the end of the chain, and the chain gets truncated because it can't grow anymore. Or what if instead of a person, you know, someone grabbed onto a tree full of sap and their hand got stuck to it, and now you can't continue that chain. That's what these dirty chemicals, these abiotic molecules, are going to do to the chemistry there. When we look at the Murchison meteorite, we see more than 10,000 different compounds there. Only a small handful of them are useful to life. And then even in silico experiments, where they use computers to simulate chemical reactions. There's a paper published a few months ago where scientists developed this computer program called alchemy. It's kind of a play on the word from the old alchemists that thought you could, you know, somehow chemically react elements together to make gold. And so this is ALL, like all chemy. And in these experiments, they'll start with, you know, 6 or so of the molecules thought to be present in the origin of life. And after one generation, you know, some of those will react, and they program this computer to analyze all the known chemical reactions of those molecules, and you get, you know, maybe a dozen or so new molecules. And then with those new molecules, you'll let them react, and then you have a second generation. So in that generation, you get even more molecules. And after about 7 generations, you get 36,685 molecules, but only 82 of those are used in life. So just after 7 generations, you have 446 times as many useless or detrimental molecules as the molecules that are needed by life. And so that's not, 7 generations isn't even going to get you to the basis of DNA. You know, you need, you know, probably 14 generations.
Let him finish, please. Let him finish.
I'm just saying,
please.
because if the sample size of if the sample size is infinite, just throwing that out there, if the sample size is infinite, you have way more than enough, you know, stuff that's going to do what you're looking for it to do out of the math you're giving. So if you're not giving a sample size, how is it even relevant? What percentage is the thing?
Because it's not unlimited. There's only so many hot little pools on Earth that you could even create some of these reactions. There's only so many meteorites that. Hold on, I'm talking. Let me finish my thought.
What about the whole Earth?
Let him finish, please. Donald, let him finish, please.
Okay.
People who are watching this want to hear both your opinion and my opinion, and if we're both talking over each other,
I just wanted to point that out so you can
and I don't have a lot of time today.
I just wanted to point that out so you can clarify, and thank you.
Okay, so it's not an unlimited supply of molecules, and even if you did have an unlimited supply of molecules, it's a probability problem. You've got, among all of these tens of thousands or hundreds of thousands, really trillions, because in this experiment they only put in molecules that we know of, and organic chemists, have identified 150 million or so organic compounds, but there's many more possibilities. Of course, most of those aren't used in life, but when we've got them outnumbering the useful ones by many thousand folds to one, then when they're assembling, the likelihood of getting not only the correct amino acid in sequence, the amino acids in sequence to form a functional protein. And yes, there's millions of functional proteins. You think about all the different enzymes and ways that they can do chemical reactions. So that there's lots of possible useful configurations or sequences, but there's many more times of just using the 20 proteinaceous amino acids that if you put them in a random order will not have a function. They will not fold into something. They will have no selective advantage where those molecules can be selected for. Now throw in all the other amino acids that have nothing to do with proteins, or other amino groups or carboxyl groups that are going to step in place of these building amino acids as they're growing, and you've compounded the problem. So Sal has a good illustration with Scrabble letters, and that's just using letters that are in the English language. This would be like, not only do you have the Scrabble letters, but some of the letters are typed on backwards. Some of the letters are in Korean, some of them are in Arabic, some of them are in Klingon, some of them are just scratches and mean nothing. And you're randomly taking letters out of that pile and trying to make words. But in that pile, there's say like 10,000 times as many of these foreign letters or backwards letters or damaged letters as the ones you actually need to make the protein. So just by probability, when you're trying to assemble those into a polymer, every one of those chances, even if you were only dealing with the left-handed 20 amino acids found in proteins, there's a 1 in 20 chance of getting the right one, or maybe less if you put in like one hydrophobic amino acid for another and it still is able to function, or depending on what your target is. If you're just trying to get a carboxylase or a decarboxylase or something, there might be several ways of doing that, but you're still talking about a very low probability of getting, let's say there's 5 possible amino acids that could go in the next position and form a potential protein, but you've got 5 out of 1,000 letters are going to be the probability of putting that right one in the next spot. So you can see, when you look at the average size of proteins today, 300 to 400 amino acids long, and maybe a protocell would get by with a shorter protein or a ribozyme or something, but there is a minimum length at which you need to have a sequence that can fold into 3-dimensional shape in order to form a enzyme-like function, a catalytic function. Okay, go ahead.
So, Sal, what would you like to add to what James said?
In forming nucleotide sequences, they have to be orderly enough so that they can be machine readable for replication, transcription, and translation. That's something that Change Tan pointed out. She is a physical organic chemist. And so the problem of having the right nucleotides available, because there are many ways to form them, where you can join the phosphate group with a nucleobase and the deoxyribose. They can form many ways, but then also when we attempt to polymerize them, and that's assuming they polymerize, because they could connect so many different ways that they don't even form a polymer. Any sort of machine, and that is a major problem. So that's in addition to just all the contaminants. The reason we know that is when Craig Ventnor was trying to make artificial synthetic life, he would have synthetic sequences that he conceived of, actually copied from a pre-existing organism, had it in a computer. He'd hire a laboratory to synthesize the sequences. One of the major problems that Dr. Tan alluded to is very similar to in the DNA synthesis. They use the blue heron process of synthesizing DNA. They can only do about 100 nucleotides at a time. The problem was they had to prevent the bonding from happening in the wrong orientation of the added nucleotide. They also had to have purified nucleotides, because if you had nucleotides that were not formed like the other ones, in terms of the deoxyribose, the phosphate groups, and the nucleosides, you could have a total mess. It's not going to be readable. So the problem, that's a slightly different problem than just the purity of the compounds, but it will affect it.
Okay, so Molecular has a question here, Saul. Can you answer
Could
to
I
that?
respond to either of them?
Yes, let's just respond to him, and then I give you the word, okay, Donald?
Okay, Sal, that's true for modern cells. How do you respond to models describing replication in the absence of extant replication machinery? Those models, like the RNA world, don't lead to cells. That's irrelevant. And there are a lot of experiments and papers like that. And it's just really not helpful. It's misleading. Not to mention that the problem, again, with the RNA world is temperature. These things have half-lives. These things can fall apart. In these replicators, you basically have already pre-synthesized huge polymers. You're not starting off with individual nucleotides, RNA nucleotides. So why cite these irrelevant experiments that don't lead to life as we know it? We're trying to solve the problem of the origin of life, not trying to make a replicator that's irrelevant. So
you just answered
citing
yourself.
those papers citing those experiments and papers are irrelevant, except for the fact they help mislead the public that they're actually making progress.
No, that's not misleading the public. And yes, they are making progress. Okay, let me go back to James first. James was ignoring natural tendencies, which make a process less random. So if you have a little pond, and it doesn't have to be a little pond, it could be a huge ocean, but if you have a little pond where you've got some chemistry going on, that chemistry is not random chemistry, because random chemistry isn't a thing. Partially random chemistry is a thing. Chemistry has tendencies because molecules have certain ways that they attract each other and repel each other, and they tend to behave in certain ways around each other. So so there's that to be taken into consideration, and then there's the scale to be taken into consideration. Are you talking about a lab sample, or are you talking about a world, a planet like this one, or many planets where maybe life will only arise on one planet out of many? And then you have, Sal was talking about these experiments that produce only one portion of what's needed to have cellular life. Well, life doesn't have to be cellular. There's no requirement for life to have cells. If you have a molecule that can be replicated in its environment, that's really all it takes. That molecule can replicate. And if that molecule can be replicated imprecisely, that means that you can get variations of that molecule. And when we're talking about, now, I wouldn't go as far as RNA for a starting point. I'd start with protein, because proteins are capable of self-catalyzing. And if you have a protein that is being imprecisely replicated, sometimes precisely, sometimes not precisely, if it's not reliable that it's going to be replicated precisely, evolution of that protein will happen over generations, and diversification of that evolution will happen. And it is inevitable. If you have enough of them, you will eventually get one that is capable of self-catalyzing, which then speeds up the process. Once you've sped up the process, then it takes a whole lot shorter time for this kind of stuff to happen. And if you have a bunch of stuff that isn't doing that, that becomes parts for the stuff that is doing that. So it doesn't take long beyond that point to have the percentages change quite drastically. And once those percentages change, then you have a tendency for something like RNA to form. It would make sense for RNA to form in that environment. So all of these experiments are just, they're finding out what tools nature is capable of using. It doesn't mean that they know which tools nature did use, because we can't directly observe the past and say, this is what happened. What we can do is we can find out what nature is capable of. And nature is very much capable of producing life. As a matter of fact, they have already produced life. Abiogenesis has been accomplished. What they haven't done is produced cellular life. yet from just plain chemistry. Now, they had produced cellular life by taking a cell and taking out the stuff, some of the stuff that makes it alive, and replacing that with other stuff that they made purely chemically in a lab. As he was mentioning Ventnor's work, where the DNA was literally put into the sequence was put into a computer, so you're not even working with chemistry anymore. And then that sequence was modified, it was stripped down, they added a signature to it, they made some other changes to it, and then they took this changed DNA and they put it into a cell that didn't have any DNA and basically booted it up, and they produced cellular life that way, from a dead cell and DNA that was produced in a lab. Not produced by a cell, by the way, not produced by life even. That was DNA that was produced from data on a computer. And so, yes, all of the steps have been done to produce cellular life, but they haven't been chained together yet. And a big factor that's really seriously important to consider is the size of the sample. When you're talking about a laboratory, yes, they have to, they have to be very precise about how they're doing things, because they have a very small sample size. There isn't really a lot of room for error if they want to have their small sample size produce results in a human lifetime, which they've done. If you're talking about, if you've got 1 billion years during which the whole planet can do this without human help, the odds are extremely high that it will happen.
Okay, James, can you answer to that, to some points made by Donald?
Yeah, so first of all, I will give you the sample size of all of the pools on all of the planets in the universe. It's going to be the same chemistry problem. All planets are going to have roughly the same elements of the periodic table, and you're always going to end up with way, way more of these interfering compounds compared to the biotic ones. What is the bond that holds nucleobases together? What is that bond? It's a phosphodiester bond. It's a bond between a hydroxyl group and a phosphate group. How many of those millions of molecules that could theoretically be made in any of these primordial soups do you think have a hydroxyl group? How many of those oceans do you think have at least a small amount of free phosphates to do that? If it's going to bind to, say, by chance, if you could even develop an adenine, guanine, cytosine, thymine, uracil, but you end up binding to some other molecule that has a phosphate bound to it, or a hydroxyl group to that phosphate, you don't get RNA, you don't get DNA, you get a little fragment of RNA or DNA plus total garbage that is not going to fold into the three-dimensional shape in order to make catalytic activity. What is the bond between two amino acids?
Why do you consider it garbage if it doesn't mimic modern life as we have it now on this planet?
Because there's a reason why. Go ahead.
We don't know how those chemicals could work with each other. What we do know is that all we need is a replicating process that is able to make some mistakes or some errors or have some variance in it. That's really all we need. It doesn't have to be anything we've ever encountered to get started. And once it gets started, it can then produce other chemistry that does likewise and lead to something like us or something like life elsewhere that isn't like us. All of this stuff you're talking about that you're calling garbage isn't garbage. That's chemistry. If you look at cellular automata, you can simplify this whole thing down. You don't even need biochemistry. You can get life-like behavior in rules as simple as Conway's game of life.
It is chemistry. It's called messy chemistry because it makes garbage. That heteropolymer with your part of the bases that we see today and part of chemicals that have nothing to do with life. So let's say you can get it to fold. Let's say you've got this mixture of biotic and abiotic molecules, and it just happened to, by some crazy chemistry, even with all these accessories sticking off of it that have nothing to do with the molecule, can form a function, like maybe it forms a bond, breaks a bond. Then what? How are you going to replicate that? That's why we have the RNA the way it is and the proteins the way they are, because they can be faithfully replicated to get that same function over and over. If you somehow could get one of these molecules to fold in solution, and you've got one of them, how long is it going to stick around until a replication mechanism that somehow recognizes this strange xenobiotic compound and can make copies of it so it can do its job again? Or do you have to wait for another molecule that can do the same job but was made up of another mishmash of biotic and abiotic molecules? You have the same thing problem with proteins. The bond that holds amino acids together is a peptide bond. It's an amide. Every time you have an amino group or a carboxyl group, they can form a peptide bond or an amide bond. So all of those millions of molecules, anywhere there's an amino group that's free or a carboxyl group, it's going to step in and junk up this fledgling polypeptide before it can ever fold into something useful. And again, you've got to be able to replicate that molecule if you want to keep using it, because the more time you add, the more it's going to degrade. Just having an abundance of water by the Le Chatelier's principle is going to cause those bonds to break. The water will hydrolyze the phosphodiester bond and it'll hydrolyze the peptide bond. You're not going to get off the ground until you can quickly reproduce these molecules.
No, that's not true. That's flat out not true.
How so?
You keep making these assumptions that it has to work like modern life and that it has to work rapidly. No, when you've got no competition out there, when it's just chemical evolution, that's all you need is the chemical evolution. Sure, it'll compete with itself, but if it's all happening slowly, then it's slow competition. And if something comes along that manages to happen quickly, that manages to do it quickly, that will outcompete. But that doesn't have to be there at the start, because that can evolve slowly out of what is happening without that. You don't need it. It doesn't need to be. It doesn't need to be consistent either. It doesn't need to be precise. As a matter of fact, it needs to be imprecise. I already said that. It needs to be imprecise. If it was precise, you'd have no evolution. If it 100% of the time replicated perfectly, then all you end up with is a bunch of the same molecule and it never goes anywhere. The imprecision is necessary for it to work. How much imprecision is a different story. Less imprecision is better, because less imprecision can outcompete the stuff that's more, well, it's better under certain circumstances. The AIDS virus, for example, is an RNA retrovirus. It mutates a heck of a lot faster than DNA does because RNA is a lot less stable. And that's one of its advantages. We have a really hard time fighting off the AIDS virus because we come up with a cure for 1 million strains of it, and there are 1 billion more strains we have to fight because it evolved that fast. I mean, it evolves fast enough you can literally watch it evolve in real time.
Do you know why RNA is unstable?
I couldn't explain it, but more or less, yeah.
Hydrolysis. Do you think life has to take place in water? Is that why NASA is spending billions of dollars to look for water on other planets?
No, I do not believe life has to take place in water. As a matter of fact, I know absolutely for a fact that life doesn't have to take place in water. Life as we know it here on this planet, yeah, it's water-based life. So yeah, this life needs it.
So you know of life that's not on this planet that doesn't require water?
I know how life works. I know how life functions. Depending on how far down. Like I said, if you're talking about cellular life, we haven't made cellular life from plain chemistry yet, but we have made life from plain chemistry. We've also made stuff that can be considered life, that fits the qualifications of life in every way except being chemical. That is not even chemistry, and that's something that I've done a lot of. And that kind of life doesn't require water because it's not even chemistry. And water is a chemical.
Yeah, you don't have to believe me. I have a PhD in biochemistry, but I challenge you to find any other abiogenesis researcher that says that we don't need water for life. These reactions take place in water. Yes, you need to dry it out in order to form condensation reactions. But again, you're only going to get these molecules to diffuse and mix with each other and interact with each other if they're in an aqueous environment. As soon as they're in that aqueous environment, they're outnumbered by, like pure water is 55 molar H2O. How much smaller are these molecules compared to that? Le Chatelier's principle is going to drive the hydrolysis reaction, the reverse reaction, and break apart polymers. You cannot have life without polymers. Would you agree? DNA is a polymer.
I wouldn't agree with that, but it's very helpful.
Complex carbohydrates are a polymer, and lipids are.
I would not agree that it's necessary, but I would say that yes, it's very, very helpful.
Then you can't have ribosomes, and you can't have proteins because they're polymers.
As far as aqueous, there are other liquids besides water that could serve the purpose under which you're using aqueous. For example, methane in its liquid form could be a basis for life as well. And that is something that, yes, if you look, you will find other scientists that are saying that methane could be a basis for life as a replacement for water under certain environments. It all depends on the environment. And we're talking about Earth's environment and not modern Earth's environment, but if we're talking about the abiogenesis that happened on Earth already, whether it was, caused by a deity or by just chemistry or whatever, that wasn't in this environment. That was in a somewhat different environment.
What is the polarity of liquid methane?
I'm not sure off the top of my head.
It's a hydrophobic molecule. It's carbon and hydrogen. They have almost identical electronegativities, so the bonds are non-polar. It makes it like a oily substance. So you're limiting severely the types of reactions can happen when you only have
I agree.
a non-polar solvent like that.
Yes,
now
I agree.
Back to the random chemistry. I agree that you would need to have some.
If I can just interject.
Sure, go ahead.
That's kind of like what I was saying a little while ago about the non-necessity of having something happen quickly, not necessarily having a catalyst that can make something happen quickly. Yeah, you're right. It's limiting, and that's not really problematic. It's just a matter of it would take more time.
And liquid methane is super cold, so all the reactions slow down too. But
yeah, and so but keep in mind that Earth, as we know it, apparently, based on the physics models, apparently Earth formed from a collision of two different planets. So the Earth-Moon system formed from the collision of two planets that were much closer to the same size as each other than the Earth and the Moon are. More of that matter went into the Earth than into the Moon, and hence the size difference. But that means that life here on this planet got started a lot later than life might have elsewhere, where such a late collision didn't happen. And so if we do have methane-based life out there, it might still not be anywhere near the stage that life here is at, but it might eventually get there, and it may have had a lot longer time already to be working on it.
Well, that's about all very speculative, but let's just talk about the RNA.
Well, I'm not speculating, I'm just pointing out possibilities. That's all.
Right,
Donald,
possibilities.
I would like to give a little bit more time to Sal talk to James, because it is really a privilege to have James here at my channel tonight, and Sal has not had the opportunity. So, Sal, can you maybe
and
ask
I want to thank
some
both of them, by the way, in
questions?
case I don't get the chance later.
Sure, yeah,
Okay.
Thank you. Sal, can I say something about the RNA replication really quick?
Please. I'd like to hear you more uninterrupted, and then I have some biochemistry questions for you.
Sure. I believe the experiment you're talking about, I'm terrible with names, but when they were looking at the self-replicating RNA strands, what they found in that paper is that the chemical selection, the functional selection in those papers and in these experiments was that the shorter RNAs began to predominate because it takes less time to replicate a short molecule. And of course, you leave the molecule, the reaction to go too long, and they just hydrolyze into pieces. So, they had to take those molecules out and analyze them before they hydrolyze. I mean, we know about the problems with the instability of RNA in aqueous solution because, look at these RNA vaccines that Moderna and Pfizer have made where they have to be kept at below -70. Part of that's because RNases that would chop them in pieces are ubiquitous, and they would also be ubiquitous in a primordial soup because along with all the favorable molecules that you would hope to get, or polymers, you're also going to get ribozymes and proteins that are detrimental, like endonucleases, exonucleases, that are going to wreak havoc and chop to pieces all of these RNAs that haven't become something useful yet, and they're trying to polymerize, and along comes this rapidly catalytic RNA that will cleave them into base pairs or a protease. I mean, there's more than 8,000 endonucleases alone that have been discovered in bacteria. That's sort of their primitive immune system to chop up viral DNA, and they protect their own DNA by methylating it so that the same enzyme doesn't work on it. So in a primordial soup, you're also going to be randomly creating proteases and endonucleases that are going to chop everything to pieces.
I actually have a paper by Steve Benner that talks about that problem, that there are more cleavers than there are connectors, and that is an acknowledged problem in the origin of life. So again, it's misleading to be advertising these RNA replicators as some sort of success without acknowledging also the fact that it's a dead end for things that Steve Benner pointed out, and this is known. He's an RNA researcher. So, but I have questions I wanted to get some clarification on with peptide chemistry. So you're saying you had the carboxyl group and then the amino group. What happens when they're interfering molecules, and are you saying that these things will be precluded from completing a peptide bond with other amino acids?
No, it's a competition. So, you have, I wish I could draw, but you have an amino group and a carboxyl group. And when they hit each other at the right velocity, the right angle, and you don't have too much water, because water tends to favor splitting them apart. But a water molecule will actually be formed through condensation reaction when an amino and a carboxyl link together to form an amide. It's a carbonyl and a nitrogen attached to each other. So that's what forms the backbone of proteins. But any other amino or carboxyl group present on any molecule, including, there's some amino acids that have amino groups and carboxyl groups as part of their functional group, like aspartic acid and glutamic acid have carboxyl groups that are what make those molecules unique from all the other amino acids. So it's not just the amino and the carboxyl that's on every one of the 20 amino acids. This is an additional carboxyl group that's part of the functional group, and that could bind to the amino and tie it up. But you also have thousands and thousands, or possibly millions. Talked to Lee Cronin yesterday, and he said there's probably trillions of possible molecules you could make with carbon. A lot of those are going to contain carboxyl groups and amino groups and other chemical functional groups that will that will be first to arrive at the growing polypeptide chain, or the growing polynucleotide chain, and get there first. So like you're hoping with a random luck that, and the laws of diffusion, that there's only a handful of possible molecules that need to go in the next in line. So the next part of the chain to have some kind of function once the chain is completed. But if another molecule gets there first by the law, of just chemical competition, and binds to the amino group or the carboxyl group first, then you've basically stopped making your polymer, or you made a polymer that's composed of part good stuff and part harmful or useless stuff that's going to interfere with the folding of the protein.
Well,
you had your hand up.
I have a couple comments on that. So
could I respond to
first
one
off,
thing
in the formation?
No, I want Sal to have some time now.
It's a really short thing. Okay, I'll just wait then.
Please,
we'll come back.
Thank you. So 25. So one of the problems is with this random formation of polypeptides, it's very hard to build symmetric like proteins, like say anything like a helicase or anything where there's mechanical symmetry. I mean, we have them in biological systems that are multimeric. The thing that helps is one has already a genome to be able to make these polypeptides. So once you form a polypeptide, the recipe's lost. There's no way to read it again and replicate it. So I just wanted to say that's moot. I did have a specific question. You were mentioning something about on other planets, what would be the probability? You were saying something about phosphate groups. Did you say there were other chemical functional groups? I missed it. And
yeah, I'm just saying with random chemistry, if you imagine primordial soups all over the universe, there's trillions of stars, and each one of the stars might have a planet in the Goldilocks zone where you have liquid water or all the elements necessary for life.
Or formaldehyde.
Yeah, it's the same. The same elements of the periodic table exist throughout the universe. So you're going to end up getting this always a preponderance of the abiotic molecules that are useless or detrimental to the chemistry. They're like chemical poisons. They're reaction dead enders, especially when you're trying to form polymers. And we're usually thinking of linear molecules because that's what they are in DNA and that's what they are in proteins. But in a primordial condition, you're not going to get a nice, like trains in a car that all link to each other head to tail, because there's also side groups. Carbon has 4 bonds it likes to form, and there can be all sorts of side reactions. This happens in our body too. Like a lot of the proteins in our body, especially on the cell receptors, they get glycated and glycosylated. Glycosylation is the intentional addition of sugars to specific amino acids on a protein to make the shape even more complex, because proteins can only make so many shapes with 20 amino acids. Glycation is what happens in your bloodstream when you're diabetic. That's when you have normally about 5% glucose in your blood. If you're diabetic, you got more than that. And even with 5%, sugars are randomly, because they're chemically sticky, are going to be attaching to our proteins in haphazard ways that cause the protein to be damaged. It leads to inflammation. It leads to, further down the road, peripheral vascular and nervous system damage. And these are bad reactions. We call them AGEs, advanced glycation end products. So this kind of messy random chemistry is happening in our bodies, and it has to be cleaned up and taken care of. And so if you have a protein trying to form, or an RNA strand in a primordial soup, you've got all sorts of other chemicals that are sticking here and piggybacking here and building this way instead of this way. And now how's this strand going to fold into a 3-dimensional shape? How is it going to be replicated when, let's say you could come up with a replication system, a simple polymerase. Well, it's used to seeing nice, clean, straight molecules to make copies of them, but now it's bumped up against something and falls off. I mean, that's how we control that's one of the ways we control polymerase activity now, is you stick our cells enzymatically stick chemical groups on there that knock off those polymerases to prevent them from continuing to do their job.
Is that what's done with the eukaryotes when they're trying to mature the mRNA? Is that what you're? I may be totally getting.
They don't chemically modify the. Yeah, it's the DNA itself gets like methylated, or the histone proteins that are wrapped around it get acetylated, and that controls whether that DNA is going to be available for replication and then transcription. So there's all sorts of different chemicals that are attached to these to DNA to regulate whether it's going to be transcribed and translated. And the mRNA regulation is a whole nother story that involves small inhibitory RNA and these RISC complexes that dicer, that you don't want RNA to be sticking around forever and making protein. You have to be able to quickly block it by making a complementary strand bind to it and targeting it to the system that quickly just chops it into pieces so the pieces can be reused. Otherwise, it would keep being translated into proteins, and you would get a bunch of proteins that you didn't need.
Now, just before one last question, the hydrolysis of RNA, I've heard about that problem. It happens. Does it happen more acutely with single-stranded RNAs?
Yeah, that's part of what makes RNA unstable, is that it's single-stranded. It gets some stability, like DNA gets some stability by being double-stranded. There are some double-stranded viruses. Their genomes are made out of double-stranded RNA that offer some stability. But there's also not RNA repair mechanisms like there are DNA repair mechanisms. So that's why HIV and coronavirus and these RNA viruses mutate so quickly. I mean, we've got thousands of different strains of COVID-19 of the SARS-CoV-2 virus just in this year because of how quickly they get damaged and mutate. But that's just talking about mutation that occurs through replication. I'm also talking about just chemical attack of the molecule, especially by water. I mean, you look at what these companies say when you have RNA or DNA, and they're like, okay, once you dissolve it in water, get rid of it after a week at refrigeration temperatures because it starts hydrolyzing, and And if there's any RNases present, which are really hard to get rid of because when you heat denature them and cool them down, they refold into the active enzyme again and will continue to chop up your RNA into pieces.
Oh, one other thing on that. In Demeter's paper, he was talking about the cycling from 0 to 100 degrees.
Right.
I was like, the half-life of the guanines is, or one of the nucleotides is
cytosine is the most
100 degrees Celsius. I'm like, did he miss that in his paper? I mean, you're going to have a disproportion. It's not, if you maintain 100 degrees Celsius, even for like a few days, you could forget one of the nucleotides.
Yeah, cytosine. That's the most unstable of the bases, of the 5 bases used in DNA and RNA.
Thanks.
Which actually isn't needed.
Thank you, Dr. Carter.
Sure.
James.
Yeah.
James, if assuming that the abiogenesis had happened at some point in time, assuming an environment similar to what most scientists who study that sort of thing think the Earth would have been like back then. If you dropped a human being back then at about the earliest stage where cellular life had started to evolve somewhere on the planet, and assuming, we're not saying the human being is necessarily put where that life started or necessarily put not where that life started, but put somewhere in the environment where life could start. How long do you think a human being would last in that environment?
What do you mean last? Until they die or until they decompose?
How long do you think a human being would be alive, or that our cell chemistry would be able to sustain itself in that environment?
Unless you have a way of metabolizing food, you're going to die.
Breathing that air, drinking that water,
Well, because when you're talking about that stage, there isn't much oxygen around, so you would die within 5 minutes.
Yeah. My point in bringing that up being that, as I mentioned, you both seem to be making a lot of assumptions about life working the way that we're used to seeing life work. Humans
but doesn't hydrolysis work no matter where you are in the universe?
Yes, the chemistry, I agree with you. The chemistry works the same no matter when you are, but what chemicals are around varies. And cytosine, is it necessary? No, it's not. It's no. It doesn't mean it's necessary. If it wasn't able to make it in a certain environment, if it kept breaking down in a certain environment, life could go on and form without it. Wouldn't be necessary, and I'm pretty sure you would agree with that. Because it is the same chemistry.
You would have to demonstrate it empirically that you can create some sort of information storage system with less bases. So you're talking about using a more robust version of cytosine, but somehow in the past that one got selected for, even though it wouldn't even have to have.
It wouldn't even have to have an information storage system. I mean, if we have one base pair, and we can make chains of that one base pair, and if they can manage to reproduce in their environment, whether they're doing it on their own or their environment itself is reproducing them by just, like I said, natural tendencies for molecules to attract certain ways. Either way, if we get replication happening, we can get more of them. And sure, we're not going to get much evolution in the sense of the sequence, but these molecules have other stuff besides the sequence. So other stuff could still end up different, the way they're folded and things like that. And in an environment that is doing that sort of replication, it's not just the replication that matters. As I mentioned, we would actually be better off without that, as far as leading toward life like we have now. We were actually better off with a replication that is not very precise at the beginning, as opposed to replication that is very precise. And so, as far as getting life started, that's a whole different ballgame compared to keeping life going as we have it now. Life as we have it now would probably not have survived long back then. Pretty much, if you dropped all of the life we have now, one piece at a time, into that environment, the vast majority of it would die pretty quickly.
And the molecules do too. They just degrade. You have no atmosphere to protect against cosmic energy. You have radioactive elements obliterating the molecules. There's oxidation reactions.
And like I said, I don't see any of that as a problem. I really don't. Is it a difficulty? Yeah. Is it going to slow things down? Yeah. Is it going to stop it? On some planets? Sure. But not necessarily on all of them. Life could still get going. And the thing is, for us to be here talking about it, it only has to have happened once. One molecule had to have been able to make it somewhere on some planet at some time in some environment.
You have a lot of faith, Donald. You have a lot of faith.
That has nothing to do with faith. That has nothing to do with faith. That's recognizing the possibilities, and it's recognizing the diversity of environments in the past and the diversity of situations that these things could have and most likely would have been happening in. And we might have ended up being being methane-based life. We might have ended up being silicon rather than
so why is it
life.
not more probable that actually an intelligent creator was involved in creating life?
Because
why
the intelligent creator,
more probable?
Because that intelligent creator would have had to have evolved first. And I'm not saying it couldn't have happened, but then we have to have an explanation for that origin as well. We don't have an explanation for the origin of the creator.
An eternal creator doesn't have to evolve. He just is.
And we also have no evidence of a creator. So there's that.
James?
Yes. It's hard to convey why this is chemically not possible. Maybe I can use a different analogy. No, it's like possible. Because you should
know that.
Your definition of life is very unorthodox. I don't see how you can have life if you don't have polymers and you don't have information. And you're talking about life without those two components. Now, any polymer is going to have to be a certain length to function, but those very molecules themselves and the bonds that hold them together are unstable. It's like trying to make the Empire, a house of cards as tall as the Empire State Building, or just a tall building, and you've got wind and you've got earthquakes and you've got things. So you might get the first or second or third level done, but no matter how much time and how many cards you have, you can't get to the length. And even if somehow it was a really calm day for a long time and you could get it to stack up, that's just one molecule. It does nothing until it can interact with the whole system of molecules. So I think you have a very, very primitive definition of life. I think life at least needs to include the ability to replicate and the ability to store information, and having a high fidelity of replication. Doesn't have to be perfect because you need that variability for chemical evolution and then biological evolution.
Are we
but
not?
I want to ask you, James, would you not say that three components
are
absolutely
discussing primitive life?
Well, Donald, can I also ask something? Because I don't want you just to concentrate on the entire conversation. I've just been listening. So, James, would you not say that three components are absolutely essential for life to start, which would be information, the basic building blocks of life, or matter and energy? And also the generation of energy is a huge problem. We need ATP, and how to make ATP. I mean, that is also an unsolved riddle. How would you answer to that, James?
I'll answer with, even if you took the simplest bacteria or simplest cell you can imagine, and it died 5 seconds ago, it's got all of the parts for life. It's got the polymers, it's got the information, it's got the molecular machines for life. But why is it dead? Because it reached chemical equilibrium. You have to be able to create some sort of disequilibrium in the ions across a membrane to power the cell. And
that does not require
the molecules that do that, the molecules with high energy transfer potential, like ATP and ADP, these molecules are incredibly unstable, and you need massive amounts of it. Your body makes your body weight in ATP every day, and then it gets a phosphate or two comes off, and then it's regenerated. So you're constantly doing a turnover of it's like if ATP was like a battery, or the battery indicator on your phone, and you see 3 bars, and you take one phosphate off, now it's 2 bars, another phosphate. You're losing potential energy there to power functions. And so you've got to be able to recharge the battery. If you don't have a recharging system, you run those molecules. Not only will they be used up, they're just sitting in water, they're incredibly unstable. Look at the product sheet, when you go purchase these nucleotide triphosphates, it says, once you've dissolved in water, throw it out after like a week or a month at refrigeration temperatures, because the phosphates come off of it. They're unstable. And trying to create those molecules requires such sophistication to regenerate them. You've got to be able to power these reactions. It's not just having energy. You have to use directed harness energy. You can't just say, oh, there's a thermal gradient between a hydrothermal vent and the cold ocean water, and that creates an energy gradient. Or even when you look at the potassium and sodium levels in the ocean, those concentrations are completely flipped of what we have in cells, because we have pumps that actively pump them against their concentration gradient. So if you don't have pumps in the membranes, if you don't have some way of creating gradients and utilizing energy, you cannot have life. You can't repair, you can't grow. You have to balance the catabolic and the anabolic reactions.
So let me ask you, James, some people say that ATP synthase was not required in order for life to start, but it could be just with fermentation and with the glycolysis metabolic pathway. What would you respond to that? Is that possible, or is ATP synthase absolutely required?
Well, there's organisms that can grow anaerobically. You have to have oxygen in order for the electron transport chain to run, in order to build up the proton motive force to power the ATP synthase. So that's something that's a very complex system. So I would agree, in their worldview, when they're trying to come up with a simple system, there are other ways of getting ATP. It's substrate level phosphorylation. You borrow a phosphate for one molecule and you stick it onto ADP and you get ATP. However, both the molecule that it got that phosphate from and the ATP itself is extremely unstable. They lose the phosphate. And phosphate isn't highly abundant in a primordial soup. Most phosphates bind to like calcium and stuff, and so it precipitates and it's unusable in solution. All right, before your head explodes.
May I please respond, because
I have not finished.
Simply.
I didn't, Donald.
Hydrothermal vent. There's your answer. Hydrothermal vent. I'm muting myself.
Explain how a hydrothermal vent gradient. You have a temperature gradient. How is that utilized? That's like saying, oh, because I can set fire to gasoline, that I can extract fuel from it.
I can't explain it because Otangelo keeps muting me and I have no opportunity to. So you explain it. You know that we need entropy. Is a hydrothermal vent, or is a hydrothermal vent not?
Donald, I didn't finish my question.
I'm muting myself.
Yes, I wanted to know, is ATP synthase absolutely required, or do we know life forms which actually do not require ATP synthase to generate energy?
That's a good question. The problem is the ATP that you get from glycolysis is so minuscule that for metabolically active cells, it's just not going to generate enough ATP. They will quickly deplete themselves of all that ATP. So, bacteria, they don't have mitochondria. But they do have the electron transport chain that is in their their cell membrane, their plasma membrane, just underneath the cell wall, and they will pump the protons in between that membrane and the cell wall, and that's parallel to our mitochondria, which have a double membrane. And those electron transport chain protein or complexes are in the inner membrane, and it pumps it in between there. So both bacteria and eukaryotic cells, prokaryotes and eukaryotes, can have this ATP synthase mechanism, but it's the problem is when the metabolic needs outpace the production of ATP by fermentation and just by glycolysis and other reactions that produce ATP. ATP is not just used as energy; it's also the substrate to make DNA. It's one of the 4 bases of DNA, one of the 4 bases of RNA. So you need ATP not just as an energy source, but you need it for as a substrate. And of course, it's also what powers thousands of reactions in your body.
Yeah, so basically it's not possible to have life without ATP synthase then. Would you agree with that?
Yes, it is.
I think it's certainly complex life. You're just not going to get enough. You get a net of like 2 ATP out of glycolysis because it costs you some in order to make it.
Yeah.
And you get like 36 to 38 ATP, depending on which organism, and if the electrons perfectly go through the electron transport chain and don't leak out. So a lot more. I mean, you also get some from the citric acid cycle, but that also requires oxygen, at least in the aerobic organisms, like
if I could,
with mitochondria.
Yeah, go ahead, Donald.
Okay, I agree with him, but with a qualification. Complex life as we know it, you would not get without it, because complex life as we know it requires it. Yes, you could get complex life without it. It wouldn't be complex life as we know it. That's all I'm saying.
Okay, Sal, you're next, please.
I just wanted to say that the problem isn't specifically cellular life. The problem is building any system that has translation, transcription, and all this integration. So we don't have to be committed, just like there are probably infinite ways of building lock and key systems and also infinite ways of building house of cards. We're not saying that life has to be built or a replicator has to be built only in this way. The issue is that we have a complex replicator with many integrated parts that are interdependent, and perhaps on some level it's more extravagant than is what is actually needed to make a replicating system. So there are, salt crystals replicate. That's it's not very sophisticated, but it doesn't. Anytime we just find a simple replicator, it doesn't mean that it will solve the problem of integration. I just highlighted some of the problems with,
and it doesn't mean it won't.
Helicases or anything that is a multimeric protein, or anything really, even a monomeric protein. If there's not a genome that stores the information so that you could repeat the recipe, all this discussion about trying to have a protein's first world with a metabolism is already moot, because there's no guarantee life is going to emerge. You're not going to have mitosis and replication and reproduction. So
you don't need a guarantee.
The problem is it's not just
and you don't need a genome.
Excuse me.
Donald, if you keep interrupting, I will have to remove you from the chat. So please respect when others are talking. Okay.
Yes,
I'll remove myself. I'm not welcome here.
Because I didn't interrupt you. You're talking, Dak.
No, Donald, you are welcome, but I really want to keep the rules, and I want to give everyone the chance to talk. I am basically not talking to give both of you the chance to have a conversation with James. He is a very welcome person here because he is a PhD. He has great knowledge, and it's an honor to have him here. So I want that both of you have the chance to talk to him. Okay?
I respect both.
Okay.
So go ahead, but you keep muting me, and I do not feel welcome, so I'm leaving. Thank you, Otangelo.
Well, you are welcome, as I said, but I don't want that people are interrupting.
So the issue is, I was talking about the complex replication cycle. So just because we can build a replicator doesn't mean we can build one as extravagant as the cell. Even I could imagine other ways to construct the cell, just reverse all the chiralities of everything. So, but it still doesn't solve the problem that you could build the cell a totally different way. That's just one illustration that we could build the cell. In fact, the fact that we have so many different architectures of cells, from eukaryotes, prokaryotes, the bacteria, the archaea, the chemotrophs, et cetera, we can implement life in a variety of ways. But it doesn't mean, in fact, I would suggest there's probably an infinite way of making replicators. It doesn't make the kind of replicators we see being highly probable, because it needs, the way that this replicator works, it has, first of all, an information storage medium like DNA. So let's just say we imagine another system. It's still going to be a problem where we have to have a reading step and some sort of translation to another chemical. Steve Benner pointed out it's very hard to build really sophisticated replicators with only one kind of biopolymer. He said RNA sorted might make it, but it will not be like the kind that we see, where there's this, like you see in a molecular, in the central dogma, where you have the DNA, RNA, and protein. And we can even imagine even more complex things. So the problem isn't making the cellular life we have so much on the Earth today. it's making life of anything of comparable complexity that has that level of integration. And just looking at a multimeric protein, I mean, that's one of my current research interests. It's like, how much has to go right to have these symmetric machines assemble and connect? So if we implemented it with, say, different materials, we would still have the same, one could envision replicators that have also these complex geometries where all the parts fit. So even like, say we built a von Neumann replicator made of silicon and metal parts or whatever, we still have the same problem of making precise 3D shapes that have to connect to each other. So whatever substrate we use or whatever chemistry, there'll still be these basic improbabilities. So just the fact that we could find these simple, maybe autocatalytic, replicators, it's misleading to suggest that it'll build something with a transcription and translation and replication system. That's just not right.
Yeah, you bring up a good point about the interaction of molecules. Sometimes I think we picture, if we can just get that one ribozyme that has, autocatalytic activity or self-replicates, or some kind of protein that will do a job. But life is the interaction of those molecules. I mean, even just forming certain types of proteins with quaternary structure, like Sal, requires the production of subunits that have to come together and interact with each other, and they have to be the right shape so that they sort of connect to each other in a physical manner. They have to have the right pockets and openings for them to interact. And then the sort of weak interactions, like the hydrophobic bonding and the van der Waals type interactions that will hold it in place long enough so that it stays there. Sometimes it'll get a disulfide bond to make a strong covalent bond. And then you get and it doesn't work until those parts come together, like the subunits of hemoglobin, or, even cholera toxin has 5 identical subunits that come together to form a ring as the part that binds to cell receptors. And then it carries the actual toxic part as a separate protein that's attached to yet another separate protein that sort of fits in that donut hole that was created. And so take one of those parts away and it no longer binds. And so every time you have an enzyme, there's a substrate it operates on. Every time you have a receptor, there's a ligand that fits just right in that pocket. And then we're not even talking about the regulation of those things, like the allosteric regulatory sites on enzymes, or the competitive inhibitors that mimic the substrate but are not turned into a product that by competition get to the active site first and slow down the production of products. And the fact that it's only the enzymes at the beginning of chains that are regulated and not the middle, because if you're in an assembly line, you don't want to just turn off one of the machines in the middle of the factory. You want to stop it at the beginning, or everything starts piling up and you waste resources if you don't operate at the junction points, the beginning of the reaction and any branching points in the reaction. And of course, that's where we find these allosterically regulated enzymes with feedback loops, feedback mechanisms that are concentration dependent.
I did want to say something, although I would totally agree, I mean, at a personal level, that ATP synthase is essential for life. I probably wouldn't make that argument in a debate. So I would, that I'm on your side of that argument, but then I just don't want to give the other side any. I think we have other arguments that we could use that are probably more forceful. And I
agree, because it's just getting to the point of where you can get the ATP synthase to operate requires so many steps before that. I mean, it doesn't operate unless you have the complexes of the electron transport chain and they have the right cofactors, and then the intermediates that carry the electrons from one complex to the other. I mean, give it to them that you can get some ATP by substrate level phosphorylation, whether it's enough ATP to live on, or their metabolism is just super slow, or they're doing these reactions in super cold water, like just above freezing, hypothetically maybe. But the ATP is it's too far along the road when they're trying to just talk about the protocell. It's way too ATP synthase, that is.
One thing that I've tried to say is, I had this exercise when I was teaching ID at college, extracurricular. And I just said to the students, I gave them a box of dice and coins. I said, just arrange it in a way that will scream design. It won't be mistaken. The way that I look at the cell, it's taking chemicals that are available on Earth, but then it's constructing it in a way that's telling you this is not the product of random accidents any more than a tornado passing through a junkyard will make a 747. It's just too well organized and connected. That's the real issue. It's not that we are forced to make life only one way. It's the fact that everything is so well connected. So there are an infinite number of ways to make aircraft, but we would still know whichever way that it's made, that there's a lot of integration of the parts. And that's more of the argument that I'm trying to make, because especially now that we have better structural data on proteins, you can visualize the improbabilities, because now we can visualize the improbability of the fold. It's so easy to see something. There's so many ways that something won't connect properly, and to have that correct fit. So I just wanted to interject that. There was one other question. I wanted to point out, Ann Gager told me, she said there may be causal circularity with ATP, that if you don't have ATP, you can't make ATP. So even with the glycolysis cycle or the ATP synthase, somewhere in the metabolic cycle, if you didn't have ATP somewhere to start to seed the system, you're not going to have any more ATP. And I thought, for the viewers here, if that's incorrect, please correct me. That's kind of an open question that some ID proponents have thrown on the table. And the spontaneous formation ATP it may happen, but it'll be in low concentrations, and I wouldn't count on it being available. I've seen some origin of life papers talking about the problem of forming ATP. Because ATP was adenosine triphosphate. So that means it needs an adenine group and a
ribose or deoxyribose,
right,
depending on
right. So you need an RNA. You need an RNA nucleotide to begin with. So it's not like it's a given that you're even going to have ATP to begin with.
In my biochemistry class, I asked the professor, I'm like, okay, so we have one monophosphate, diphosphate, triphosphate, 1, 2, or 3 phosphates. I said, are there tetraphosphates or pentaphosphates? And he's like, well, they're rare and they tend to cyclize, but they're just too unstable. Even having 3 phosphate bonds next to each other, you have all those, depending on the pH that it's in, it ionizes and creates a bunch of negative charges that repel each other. And so that phosphodiester bond in the DNA and RNA will cleave just spontaneously rather easily. But yeah, just having the 3 phosphates back to back are unstable because those linkages are under some stress there. Oh, and the costing ATP in order to make ATP, I think that's referring to glycolysis. There's 10 steps and there are 10 enzymes that convert glucose into 2 molecules of pyruvate, and you end up making 4 ATP in that process, but 2 of the steps are energetically unfavorable, so it costs 2 ATP. So it's like you got to spend $4 and then you get $2 out of it. So is that 2 ATP going to be enough to live on, especially when you can't keep it forever? It's not like you can stockpile ATP somewhere because it's just too unstable.
And so I think the point was, this is also like the other chicken and eggs paradoxes that are a little bit more subtle. That people, there's so many of these. If someone were just willing to catalog them, we've only, I think, scratched the surface. I think Ann Gager pointed out there's certain amino acids, if you don't have them, none of the metabolic cycles would be able to make them. So there are other chicken eggs paradoxes with the synthesis of amino acids. You need that amino acid to make other amino acids.
Oh yeah, you need proteins to make DNA. You need DNA to code for the proteins. You need proteins to process the other proteins. You need chaperones to help make sure the proteins fold properly. You need proteins to cut off, like components that are needed to code the direction of where these things end up and then cut them off later. I mean, there's just so much modification that goes on that I think people just have no clue about the how all of this is coordinated and regulated, and how it all interacts with each other, and just the sheer number of interactions by different molecules that are needed just to get the basic, like the foundational life going.
I am studying about the biogenesis of the ribosome, James, and I think it is the most complex thing that I have studied in regards of biochemistry so far. And there are some science papers, and at each second sentence they put in regards of something a number which is a link to another science paper which is just about that sub part of the ribosome. And I am just now starting to scratch at the surface of how this thing goes. And I am at that moment now completely lost. I mean, there are so many technical terms that I have to learn them before that I even can start to have a superficial grasp of that. But I mean, there are so many things that you see. Well, there has to be everything there, otherwise it doesn't work. Like, for example, when a ribosomal RNA comes out of the ribosome and is synthesized, then a chaperone immediately comes and binds it. Otherwise, it will fold, and it cannot fold at that stage. So it prevents it to fold, and it carries it to another department where then it is tested. It's literally tested if it is okay, if it is a functional part. And if it's not, then it is discarded in the proteasome. But if it is functional, then it is integrated in the assembly process of one of the two subparts. So this is high engineering. And if that has to come before life started, how do you get that kind of sophistication without evolution or without intelligence? I mean, this is completely impossible in my understanding.
Yeah, in graduate school, one of my professors did his PhD research just on the ribosome, and it's, yeah, you know, they talk about ribozyme activity, and well, could it act as a sort of translational mechanism? But because the ribosomal RNA is what is actually catalyzing the linkage of the amino acids together to make proteins, but it can't do that without the ribosome acting as a scaffolding, as this, you know, there's the large subunit and the small subunit attached to the messenger RNA, but the ribosomal RNA that's woven into that protein of the ribosome has to be, I mean, basically that's the job of the protein, is just to bend the RNA, the ribosomal RNA, in just the right shape and orientation so that it can have enzymatic activity, so it can help form these peptide bonds between the amino acids. And I'm still learning stuff about the translation process. I just found out, you know, with all the different tRNAs and how there's a different one, you know, you have to have an amino acid attached to it, and then the anticodon sequence has to match, you know, the codon and the mRNA. Well, I knew there was an enzyme that was unique for each of those amino acids and tRNAs, the aminoacyl tRNA synthetases, that has to be able to recognize, okay, alanine is going to be attached to this particular tRNA, but not any of the other, you know, 64 tRNAs. But that's not the only enzyme involved. I guess there's like a dozen other enzymes that are involved in just prepping that tRNA molecule to get it ready for action.
Yeah, and the amino acid has to be charged by the aminoacyl tRNA. And I mean, it's such, and it's such a complex process. And I mean, naturalists, they think that it can, that machine can form by a stepwise evolutionary process. But I mean, I cannot imagine how that could be. If you imagine that just the peptidyl transferase center, the part is a ribosomal RNA, and it has about 2,900 RNAs linked together. How do you get that in the right configuration to have that center?
And what if the little loop that forms the anticodon sequence wasn't bent at just the right angle, so that there was only 3 bases on the anticodon to match up with the 3 on the codon? Like then you, instead of having a a triplet codon, you might have a duplicate, 2 or 4 or 5, and it's got to be consistently 3 every time. Otherwise, you'll have frame shifts and get a totally different signal.
And that's why I like all the visualizations. You can actually see the improbability based, rather than just saying, a vague fold of an RNA or whatever.
Like a proto-RNA, tRNA, that's just randomly forming in a primordial soup.
You can actually see the improbability based on the shape and what it has to connect to. It's just like a nut fitting into a bolt. Royal Truman mentioned that even some of these folds in the tRNA are not possible unless there were RNA modifications. So the term is like A6 amers. I don't know what they call it, an epitranscriptome modification. And you actually need proteins to make that modification. Otherwise,
sometimes they'll go through chemical modifications too. Not sometimes, but often.
It won't have that fold. It needs that modification. That modification has to be on the right nucleotide, and that's not easy to do. To get it to give it that right fold.
Be that selective.
Yeah. So
you don't want to just randomly stick chemicals onto other molecules that are similar. It has to be very precise in its recognition of that molecule.
Before we close the show, and I don't know how long I'll be on, I just wanted to publicly apologize to Otangelo. I put him in a hard spot because I was complaining in the private chat that I didn't want Donald here. Not so much because I didn't want him to have his voice, but it's just so rare to have a biochemist talk, and so I was a little bit rough on Otangelo, saying we need more time with Dr. Carter. So I just wanted to say I'm sorry, Otangelo. I didn't mean to cause trouble for you, but I was interested in the quality of your show and our having ID proponents who are qualified. Qualified like Dr. Carter.
I really don't want that atheists think that they are not welcome at my channel. They are welcome. And Donald is also welcome. He said, I'm leaving because I do not feel welcome here. And I want to make that very clear. He is welcome as any atheist to come here to my channel. And I want to have polite conversations with anyone of any worldview. Now, what really was difficult is to contain him. He started to gesticulate and so on. And if it's not his turn, then just keep listening and do not try constantly to intervene and to interrupt. And that was really a little bit my concern. And I wanted to give you, Sal, also the time to have your conversation with James. So I said to him previously, before he came to my show, I said, Donald, we had a conversation previously and you were interrupting me and I left because of that. So you are welcome to come, but please do not interrupt. And he was simply not able to keep to my terms. So he is welcome to come again, but I really want polite conversations here and without interrupting.
So I'm really glad that you came, Dr. Carter. What ends up happening is sometimes the environment can be so hostile that scholars just don't even want to come to the internet. And
I've been following you guys for long enough. I know the different personalities, and I totally do not mind. In fact, I encourage opposing viewpoints because that helps us sharpen our own and make sure, I didn't think about that, or that's a good point, and hopefully we can do that the same. But what I don't tolerate is the constant interruptions because it wastes everyone's time.
That was my concern.
Whether they agree with me or whether they agree with him, I want to hear what he has to say. That's why I don't interrupt him, because I want to see if there's anything that I can pick up on, or I need to be paying attention so I can give my counter response. And so when you're talking over each other, it's just wasting everyone's time. I'm taking time away from my family and kids to do this. I don't want to just be extending this another 20% by having to repeat myself.
And I'm glad, I do listen to what the other side says. There were some good points that the other side said that I felt had to be addressed in my debate last night. I felt everyone liked the way that it was conducted. I thought both sides were representing themselves well as human beings, and they're polite and civil, and that's more the way that I like communication. So I just wanted to publicly apologize to you, Tangelo. So
no concern. I think it was important for me to say what my viewpoint is on this. That Donald Dussey said, "Oh, I don't feel welcome here," or "I am not welcome here." No, he is welcome as any atheist, as anyone, but really, anyone should simply not interrupt and let the other side talk. That's really what concerns me.
I do think that, smaller groups, smaller panels, maybe of 2 or 3 work. When we have like 5 people on, it just starts to become unmanageable.
Yeah,
so
I agree with you.
My sincere apologies to Rational Empiricist. I called him some not so nice names, and I'm sorry to you, Rational Empiricist. I was just getting upset because these guys are on the internet a lot, on Speed of Sound's channel and these other atheist channels. It's just like, we don't need a replay of that on the Daily ID show. That's just my feeling, but it's not my channel. And if I were stepping out of line, I apologize for that too. But part of it was, I'm really so glad to see Dr. Carter here. It's been
I too.
years since we've had a biochemist, a professional biochemist willing to interact with us, because we have all these non-professionals talking about chemistry, and I'm learning chemistry. And finally, we actually get someone who actually teaches chemistry.
It's so hard to do this without illustrations and trying to describe stuff. And I'm throwing out terms, and I'm like, oh man, I probably just lost like 90% of the people listening because they don't know what that term is. So in the future, this is sort of an impromptu thing. Like a couple hours ago, we're like, hey, let's talk about this. And I said, oh, that sounds interesting. I'll join. And so maybe, I want these to be as informative as possible and not to go over people's heads. So, when we talk about, a new paper that came out, or we discuss the pros and cons of something, to have it be, efficient and learning experience for everyone, us included, then we're not just wasting gigabytes on YouTube and everyone's time.
Yes, and I mean, there were some interesting comments by Molecular Alchemy, and he's talking about like a five nucleotide ribozyme, and I thought that was interesting. It's like, well, I think it's kind of irrelevant, but it was still kind of interesting.
The function of that would be so severely limited, because unless you can you have to have enough loops of RNA or proteins to fold into a secondary, tertiary, quaternary shape, to create pockets, to create loops, to create grooves, in order to secure a molecule for it to work upon, to chemically react with. It's got to hold on to it. So that requires just a certain a length of your polymer to be able to form all of these little nooks and crannies and pockets. And so with just 5 nucleotides, I mean, how are you even going to be very selective in the type of chemical reaction that's occurring there? It's just going to start randomly doing something to a half dozen or hundreds of different similar looking molecules.
See, I'm glad I put that on the table. See, now that kind of puts that one to rest. And this is what I see is they'll cite something like that and then give the impression that, oh, therefore all this can naturally evolve. And that's kind of been my complaint with a lot of these papers. I said, yeah, a tornado can pass through the junkyard. It can make a lot of paperweights and primitive hammers. It doesn't mean it's going to make a 747. So a lot of these citations of random variety polymers, be they nucleotides or amino acids, and they say, oh, we found it had some use. It's kind of misleading to suggest then that you could build something really complex. It'd be like me saying, oh, look, a tornado passed through a junkyard. Look at all the paperweights it made. Therefore, we can make a 747 with the same process. That's just not, I have problems with that.
Well, in nature, you look at what is the shortest ribozyme. And last time I looked, it was the hammerhead ribozyme, this RNA that folds into a secondary structure that has enzyme function. And I think it's, I tried to look at the sequence, and it looks like it's actually a conglomerate of several strands of a couple dozen nucleobases long each. So, you're still talking about a few dozen long to get a minimum enzymatic function out of an RNA. And then it's very challenging to try to replicate let's say you could get it. Once it's folded like that and it's hydrogen, if you have a strand like this and it folds back on itself and starts hydrogen bonding right here to form a loop, and this loop, plays some sort of chemical role in whatever it does, trying to replicate that molecule, you need to straighten it out again. You need to, unless you have an enzyme that can break those hydrogen bonds and make the molecule linear again, you've got to heat it up to like 95 degrees Celsius to melt apart all those hydrogen bonds. The covalent bonds that hold the bases together are strong enough to survive that heat, but all of the numerous hydrogen bonds that help make these loops and these folds are going to get in the way of any kind of replication system. And again, like in the experiments, the RNAs just get shorter and shorter because we're talking about chemical selection, and it's favoring whatever molecule reproduces the fastest, which is going to be the smallest molecule. We need functional selection, which is going to have a purpose that is then somehow retained over all the other molecules that don't have a function.
That's actually one of the most well said things I've heard, is that natural selection is not functional selection. If I recall,
it is at the biological evolution level. They would say, okay, you're selecting for molecules that confer a reproductive advantage, but I'm talking about at the primordial, the abiogenesis level. We don't have that. You don't have replication. You can't have evolutionary selection or natural selection until you've got faithful reproduction of that system. So I'm talking about molecules that are just randomly forming in a primordial soup. They're going to be chemically selected for. Right. Even if you could stabilize them with peptide RNA link, couplings, you're only going to be selecting for that which is the most like thermally stable, or that's based on the preponderance of the various building blocks, and whether they agree with each other or not, whether they I'm not saying every reaction is possible, but in that pool of molecules, there's going to be a certain percentage of them that can react, depending on, some of them are going to repel each other. Some of them are going to cause chemical reactions and change into a new compound. But those, you're not selecting for the molecule based on whether it can, say, form bonds or that has some kind of enzyme activity, like transfer an amino group from one molecule to another, or hydrolyze a molecule, or form double bonds. Those kind of functions that you need for life. You're not going to be selecting for those. You're only going to be selecting for whatever happens to fit nicely next to its partner, and whatever happened to be in higher concentration in the solution, and whatever got there first, and whatever survives higher temperatures. Like, you're going to end up with RNA with more G's and C's in them than A's and T's, because guanine and cytosine form 3 hydrogen bonds, and adenine and thymine only form 2. So you would be selecting for RNA that's more thermally stable, that would be richer in G's and C's, which is going to limit your variability in the code, because now you've cut out the percentage of A's and T's to create variability, to create variety.
And what you have said that's the money quote. I'll try to remember it. Yep. Chemical selection is actually the true natural selection. What the
chemical and physical selection, like,
right.
Temperature gradients, hot cold cycles,
right.
Wet dry cycles,
chemical and physical. Whereas what some of the mainstream call natural selection is only fantasized. It's not what really happens in nature. They say, well, natural selection, they'll just say it's going to be functional. It's like, that's just imaginary, because there's no basis of that in chemistry and physics. There was one other thing I wanted to say, and now I'm getting really forgetful in my old age. I had to wait.
It's Friday afternoon. Yeah.
If I can, it had something to do with, as you were talking about you were talking about the chemicals, and now it just slips my mind. But I think it had something to do with functional selection. We can't. Oh, now I remember.
Yay.
We can't. It's false advertising to say, oh, we just created a catalytic reaction, and now They may call it enzymatic. Therefore, we can make other reactions, that's a false extrapolation. So let's say they modified some enzyme, just some monomeric enzyme, and it can now catalyze a new reaction. It's not appropriate to extrapolate that to something that is for multimeric enzymes. I mean, that's just ridiculous. Just because you have a catalytic reaction, say even with a 15 polypeptide polymer or 15 mer polypeptide, just because you found a catalytic reaction, there's no way that should be legitimately extrapolated to say something like a helicase or topoisomerase or a polymerase will evolve naturally. But I see that kind of that insinuation all the time. Oh, because we were able to do it in small pieces, we can do it incrementally. And it's like, no, that's not going to work for a multimeric enzyme. The multimeric enzyme only works when you have all the pieces together, and usually they're very large polypeptides and at least dimers. But then we see some that are big. I saw some of the ones in the like say metabolic cycles. They're huge. Their quaternary structure is huge. So I just wanted to say that it's illegitimate to allow, just like when molecular alchemy said, look at this little 5 nucleotide ribozyme. I just kind of brushed him off, but I didn't justify what I wasn't backing it up because I didn't have time. But now looking back, I said, that does not justify the emergence of multimeric enzymes. It totally fails as an explanation that a multimeric enzyme will succeed, because if you just have one part of that, one polypeptide floating around, it might catalyze something, but what really makes it work is when it's able to join with its partner. And I don't think that can be built incrementally.
Yeah, claiming victory because you found a 5 nucleotide long catalytic function is like saying, well, we got to build some kind of house or structure or some way of providing food and shelter, and you found a little piece of Scotch tape like this. There's a long way to go before you have a shelter with a little piece of tape. The other thing is, just because you have something catalytic does not equal good or beneficial, because catalytic just means it speeds up chemical reactions. But what if that chemical reaction is bad? What if that chemical reaction is, I'm going to chop everything with a peptide bond into fragments? Like water, let's say you're sawing down trees, and the saw cutting down a tree takes a long time. Water just being there, just by the laws of it randomly bumping into these peptide bonds or these phosphodiester bonds in the DNA and the proteins, it's going to split it without any sort of catalytic activity. It's just that it's a reversible reaction. Condensation and hydrolysis are reversible reactions. So condensation's needed to take 2 monomers and link them together. Whether you're talking about amino acids linking together to make polypeptides that then fold into proteins, or whether you're talking about the bases, the nucleotides, and getting them to link together, water will break those apart. But there's many, many enzymes. There's endonucleases that will cut in the middle strands, and there's exonucleases that cleave off from the ends and work towards the middle to chop up RNA as well as DNA. There's endonucleases for both of those. And through random chemistry, if you're hoping to get catalytic function, you're also going to get the catalytic ability to speed up the degradation of all your precious RNA and DNA in that random primordial soup. Same thing with proteins. There are many different types of enzymes that chop proteins to pieces. You might be thinking, well, how come our cells, if we have these enzymes, why aren't they chopping us to pieces? Well, when you eat protein, for instance, you create 2 of those proteins as zymogens. They're precursor enzymes, kind of like it's like keeping the axe in a sleeve so it can't do any cutting. And then when you eat food and the stomach acid starts being produced, the chymotrypsin and pepsinogen in their inactive forms become activated when the hydrochloric acid starts being pumped into the stomach. So then you get pepsin and trypsin, and then they can start chopping up proteins into pieces so that you can reuse those amino acids as fuel or to build new things in your body. So they're not active all the time, and they're only active in certain compartments. And so these proteolytic enzymes are heavily regulated so that they don't just go chopping everything up that you work so hard to create. So there's a whole component of regulation to that that's involved. So in the same kind of random chemistry, this is why I asked the question in your debate last night, Sal, is how are you going to control or prevent these polymers that have, whether it's a ribozyme or whether it's an enzyme, that are going to, how are you going to keep them from chopping all your valuable stuff into pieces unregulated? Because it will, I mean, some of these enzymes will cut, 1,000 molecules per minute or per second. They're incredibly fast. So instead of having a saw to cut down trees, now you've got a gas-powered chainsaw, and you have no off button and an unlimited supply of gasoline. I mean, you're just going to cut the entire forest down, and that kind of stuff has to be regulated. And so if you take away what power, some of those enzymes don't even require ATP either. They will, all they need is like a free hydroxyl group, and they will cut off a base pair. And some of them do take up ATP, and if you run out of ATP, okay, that slows down the enzyme. Or if you run out of the cofactor that enzyme needs, that will slow it down. But that's not how our cells regulate it. It regulates it through inhibitors and all the mechanisms of stop making more enzymes. And then there's other mechanisms that will break down those enzymes so they don't work anymore. So there's so many levels of control and regulation all along the way, from assembly of these enzymes to the degradation of the enzymes to pre-packaging the enzymes in an inactive form or compartmentalizing them in a vesicle so that they can be released all of a sudden and do a quick job instead of waiting for them to be transcribed and translated. But in a primordial soup condition, you don't have all those regulatory mechanisms. You're going to just as likely get a bond-breaking molecule that is going to wreak havoc on all of the molecules in the vicinity that have that bond that it targets.
And you did point out something interesting, and I actually have another meeting. I have a regular Friday night meeting.
I got to get going too, yeah.
You talked about water being a problem, and Steve Benner pointed out that's the Origin of Life community knows this, not just Steve Benner. They call it the water paradox.
Yeah.
You can't build life with water, and you can't build life without it.
Yep.
And so they're proposing formamide, an earth covered with formamide. That's how we were able to solve. It's just like, yeah, you just created a whole nother set of problems.
Yeah, so you're creating cross-linking molecules that are going to turn everything into a giant ball of tangled garbage, which we call tar or asphalt. And the other thing they'll say is, okay, well, if you have wet dry cycles, like an area where a thermal pool, drops down and then the molecules dry out a little bit, then you can prevent some of the hydrolysis. But so what? Then you've got a molecule that's dried out, stuck to the rock, and the only way for it to participate in any other reactions is to get back in the pool where it can serve a function. And as soon as it gets back in the water's waiting there to kill it again.
Yeah. So all this to say, the designer assembled cellular life in a way that just says, no, this isn't going to happen naturally. That's what he could have chosen another way to do it.
I think you are just responding to Top Dog here.
Just because an intelligence can make things doesn't mean nature can't make things naturally. Yeah, but can it do it with specificity? I mean, sure, snowflakes can form, salt crystals can form, but and you can even use catalytic surfaces to replicate things, but it lacks the sophisticated diversity that you need to, in other words, the information, the coding in there to form novel functions, and then you got to be able to replicate it faithfully.
Right. And it's the organization. And yesterday, just briefly before we go, Immutable Destiny said it's just chemistry. There's no need for intelligence. I wanted to say that's obfuscation and equivocation. No one in the ID community is saying, oh, the way a cell works in normal operation, intelligence is constantly present. That's kind of an insinuation that isn't correct. But what I wanted to say is we're not talking about the operation, we're talking about the origin. And it's just like you could say a chemical factory, it's just chemistry that makes it work. I'm just like, well, it makes it operate, chemistry and physics, but the origin, the organization of it had to be planned by intelligence, because you have all these step reactions, and the cell is a chemical factory. That's the issue. Where did the source of the organization come from? It doesn't happen spontaneously. And I really just didn't have time to deal with Immutable Destiny's claim, but that's the thing that Speed of Sound and Immutable Destiny keeps saying, oh, it's just chemistry. I'm just like,
they don't get it.
That's obfuscating the issue. Yeah.
Well, it's like having a bunch of wind-up toys, and like, sure, once you wind them up and you let them go, look, it's operating all by itself through totally natural processes. You know, that spring is releasing the tension and stuff. It's like, well, okay, well, something had to come along to wind up that toy. Something had to design the toy to the parts interact with each other. And
as a last word, James, I would just like a confirmation about something which I commonly say to atheists, and getting the confirmation from you, that would be just reinforcing what I constantly say. For a long time, I believed that the smallest living creature would be Mycoplasma genitalum, which is about 500,000 nucleotides big. And then about 6 months ago or so, I discovered actually that this is a pathogen which requires a host to survive, and it doesn't make amino acids by itself. So I said,
that's why it's a sexually transmitted disease.
And you cannot use it as a model to say that is the smallest living life form, and probably that would be the size of the last universal common ancestor. So I went to search what would be the smallest free living bacteria, which makes all the basic building blocks, including amino acids. And the smallest I did find out is Pelagibacter ubique. That is a bacteria which uses about 1.3 million nucleotides. So I made a calculation, and in order for you to have the minimal proteome with that size of a genome, you require a chance of 1 to 10 to the 722,000 of power. So
that's if you're only starting with sugars that are in the right-handed configuration for the ribose and amino acids that are in the left-handed, not all of the tens of thousands of xenobiotic and abiotic molecules that would interfere with that. And the other thing is being able to replicate that faithfully. I mean, the DNA polymerase that makes faithful copies of that genome has an editing ability that reduces the error rate to an incredibly low number. You can knock off that part or knock out that part of the editase component of the polymerase, and the number of mutations makes that organism reach error catastrophe incredibly fast. I know evolutionists want to say, well, you need to have some randomness, because that's, it's the random formation of differences that get selected for through environmental pressures. But there's a very narrow, I'd say really non-existent sweet spot between high fidelity replication and allowing it in enough errors so that you can have variety and variability. But to give you an idea of how quickly error catastrophe can occur, in one of my undergraduate microbiology classes, we grew these strains of E. coli that had a mutation in just one of their repair mechanisms for ultraviolet induced DNA damage. So normally DNA, goes through, thousands, if not millions of mutations per day that have to be repaired. And one of those is caused by ultraviolet light. Like if two thymines are next to each other, they form this thymine dimer that makes a kink in the DNA, and then the polymerase gets jammed against it. So there's a whole system of enzymes. Even in E. coli, there's like at least three different repair systems for fixing problems like that, where the DNA gets damaged by ultraviolet light. So just one of those genes was knocked out or damaged and not functioning. And we grew plates of them both in the dark in a drawer and then on the windowsill. And within a matter of days, every single one of the billions of cells that were in that petri dish died. There was no new colonies that would grow because they had acquired so many mutations from ultraviolet light that they reached error catastrophe. Their genomes were so flooded with mistakes, or the polymerases weren't able to slide across the DNA and make copies. And then, of course, the RNA polymerase couldn't make its mRNA, and then you couldn't make proteins, and then the organism dies. Can't make ATP, can't do all its enzyme functions.
That's why I say life is an all or nothing business. You have to have everything together, otherwise nothing goes. If you don't have the repair mechanisms right from the go, then happens exactly what you said right now. And what amazed me in regards of the ribosome, I knew that the ribosome has about 11 or 13 different error check and repair mechanisms during the translation. But when I read that even the assembly process, each single protein is a test drive. It makes a test drive if it is proper, and only the ones which are correctly synthesized are incorporated into the end product and the end assembly. So you have error check during the assembly of the ribosome and error check during the working of the ribosome. If that is not the evidence of design, then I don't know what is.
That is so important too. There's not just error checking when you're copying the genome, and there's also editing with the assembly of the protein. And if there's a problem, you also have to have the ability to clean up that mess. When I was genetically engineering E. coli to produce human insulin, some of the things I was genetically engineering them with, it made proteins that the cell just couldn't handle. It couldn't fold it, and so it would cause a problem. So it would, bacteria don't have lysosomes to chop all these up. They have they wouldn't have lysosomes because those are organelles, but they do have proteolytic enzymes to help clean up this mess. But usually what they do is they have like a trash heap that they take all these misfolded proteins and clump them together in these inclusion bodies, and you can see them in some cells in a microscope, but it has to be able to deal with misfolded proteins. And so our cells have all sorts of enzymes for either transporting and sequestering them into a compartment where they can later be broken down and degraded and recycled and reuse the parts, or they wreak havoc. It's like throwing a monkey wrench into a very sophisticated machine, and it that could do some serious damage. A loose bolt in a giant factory machine could make everything screech to a halt. So you've got to make sure that there aren't just scraps sitting around that are going to cause problems. Cell's very efficient at cleaning those up.
I've been using that argument, and I said, if you do not have the proteasome, which cleans up the mess, if an RNA strand is misfolded and is not working properly, these misfolded proteins, they will start to accumulate in the cell, and in the end, I think that the cell would die. Is that correct?
It can, yeah. It can be lethal to the cell. Happens in humans too. When there's a common genetic disorder where you don't make enough of your own antioxidant called glutathione, so free radicals damage your hemoglobin, and it clumps together in these Heinz bodies in your red blood cells, and makes you resistant to malaria. That's why it's so common. This glucose 6-phosphate dehydrogenase mutation, but it does lead to cell death. Those red blood cells have to be cleaned up, and the spleen, and this accumulated damaged protein can be lethal to the cell.
Yeah, we are close to 2 hours. I would like to give Sal the last word, and then you, James, and we will close our stream. So if you have one more question, Sal, you can ask, and then I think we can end this stream here.
I just have a quick comment. Dr. Carter, you inspired me to keep studying chemistry, and I will learn it by trying to teach it and teach Dr. Tan's molecular biology course. So thank you. You're an inspiration to all of us, and I hope some of the viewers will be inspired to study chemistry, because these are more the level of discussions I feel will move forward the intelligent design argument. when we get into these levels of details, it's much harder for the other side to put forward their case. They like very ambiguous, vague things. So when you start to get into the specific chemistry and the amount of organization needed, the idea argument becomes far more forceful. So thank you for giving us all these facts. And it's just a pleasure to just sit here and learn, and also teach me all the things I need to learn. And
me too.
Extremely grateful.
Yeah, thanks, Sal. I agree that we really got to get down to the nuts and bolts of understanding these pathways and mechanisms. And unfortunately, you do have to have enough of a background in chemistry and biochemistry, molecular biology, cell biology, to even understand the terms that we're talking about. So that's why I'm grateful that I think as people start to listen to these talks, even if they're like, oh, what is that? That's a word I don't know. Let me look that up, and they'll find a cool animation or video and begin to understand. I've been doing that. I've been learning new terms by listening to your guys' channels, and I'm like, oh yeah, I can't remember the details of that. Let me look that up again, and then I come back next time even more prepared to be able to comprehend what the conversation's about. So I know there's some people listening that are like, wow, this is all over my head, and so I encourage you to check out Sal's Reasons Academy. What's the website again? It's
Evidence Reasons Academy. They can find it just by going to my website, evidenceandreasons.org, and there'll be links to the academy. I only have 5 chemistry lectures out. I'm going to have just for general chemistry 240 lectures. 120 will be the actual lecture because they're short, like 30 minutes, and then 120 lectures that are walking through the homework exercises. Just for, there are other ways you could learn it. There's the MIT courseware and stuff, but I decided that I needed to improve my professional effectiveness by learning a lot of the classes I've forgotten the material. And one way to learn it is to teach it. And I said, if I make it a video, I could also do a service to our community. So maybe if they learn nothing else, they'll see that, well, sometimes you just have to slug through things that might put you to sleep. And I have to make a confession. Sometimes when I've listened to my own lectures, it put me to sleep. So I take no offense if people are bored. If you struggle through it, more power to you. And I'm there just to serve you all.
Yes.
It's a challenge when you're teaching, because you have to be able to read students' faces to see if they're sleeping or they have a bewildered look on their face. And when you're on YouTube, you can't always know what's going through people's heads. And so it is a challenge, but encourage everyone to just keep on learning. The knowledge is power. I was the same way there. I took all these classes for 27 years. I took classes, and half of that was science classes. And now I'm teaching it, and a lot of it, like I've taught over 15 science classes in the last 10 years. And there's times where I'm like reading the book the night before I'm about to teach it, like, oh yeah, I totally forgot about this, or I never really understood that part of chemistry. And so, yeah, it helps definitely sharpen my skill set by going back and having to teach it and relearn a lot of these things.
Yeah, what I am learning is that we never stop to learn, and this, if you start to understand and biochemistry, then you will see that the universe about this world is so huge that even if we studied for our entire life, what we have learned compared to what is out there is just a tiny fraction, and I think that keeps us humble. But James, what I special appreciate about you coming to my channel and interacting with us is that I am constantly looking at atheist channels, and there is people out there. I had interactions with PhDs, and they are criticizing me and say, oh, Tangelo, he's not learning because he had interactions with PhDs, with that guy and with that guy, and he still doesn't get it. And tonight I have someone here at my channel, you, James, who are also a PhD, and I can say I sign below everything which you said tonight, and I am fully in line with what you are saying. So
good. That means you understood what I was saying.
No, I didn't understand everything, but okay. we are on the same side of in the same boat, let's say like that. So if these guys are criticizing me that I have an inferior understanding than PhDs, which are professional, but I am agreeing fully with what you are saying, and you have also a diploma in PhD, so then they cannot continue to criticize me that I am wrong because of that.
Don't let them lord over that PhD with you. You don't have to have a PhD to be highly knowledgeable. I mean, in fact, usually when you get a PhD, you just become more narrow in your focus, and you sort of specialize in one little area, and then everything else kind of becomes a distant, foggy memory. So yeah, it's certainly, it takes a lot of work. I don't think I could go and do it again if I knew how bad it was going to be, how tough it was to get a PhD. It took me 9 years, but partly because my research was so sophisticated, and some of the experiments took a long time. I had these 16-hour long experiments that were just brutal, and but it's don't let that discourage you. I listen to people on YouTube all the time that don't have PhDs, and I feel like, wow, they are really bright. They have a higher IQ than I do. They know a lot about this subject, and I can learn from them. And so you got to be humble about what you know. We don't always know what we don't know either, and the way you find out is by engaging in people like we're doing now.
Yeah,
and reading papers.
I think the more that we actually learn, the more humble it makes us compared to that amazing creator which made it all. And we try just to be light in a world which dwells in darkness, and pointing to that amazing creator. I mean, it's not about us, it is about God. It's about the one which made it all. And we are just tiny little creatures which try to understand just a fraction of what he did, and which points to him. But okay, so I want to end this stream. We are 2 hours here. James, once again, thank you very much for coming to my channel, and I hope that we can have more of such interactions. And I think it's very beneficial because these streams, they will remain online for many years, and people, they will listen to them and learn. So if you have time, I encourage you to keep doing this. We have not many PhDs and highly educated Christians which are having the courage to come public and teach us. So if you can keep doing that, then I would want strongly to encourage you to do it at Sal's channels, eventually at mine, at other channels. So once again, thank you very much. Thank you very much for everyone which has assisted to us. Until next time. Bye-bye.
Thank you. Bye-bye.